The overall goal of this procedure is to culture human umbilical vein endothelial cells and to analyze cytoskeletal dynamics and cell migration using live cell time-lapse imaging.
Methods described in this video can help answer key questions in the field of cell, molecular, and mechanobiology. For example, this protocol can be used to identify how cell shape and motility are modulated by engagement of different ECMs.
Visual demonstration is critical because the sample preparation steps are difficult to perform. They require precise handling of the cells in a timely manner throughout the process.
Though this method can provide insight into endothelial cell motility during angiogenesis, it can also be applied to microscopic investigations of virtually any adherent cell model system.
Using forceps, pick up a cover slip from a 100% ethanol storage jar and pass it through a Bunsen burner flame to sterilize it. Then place it in a 35 mm Petri dish. Prepare one cover slip for each sample. To a 1.5 milliliter microcentrifuge tube, add 10 microliters of 1 milligram per milliliter fibronectin stock solution and 990 microliters of PBS. Then pipette 250 microliters of the solution onto each cover slip. Spread the solution evenly to cover the surface. Incubate the cover slips for a minimum of 3 hours at 37 degrees Celsius. Before use, rinse each cover slip 3 times with 2 milliliters.